DNA sequencing is the problem of figuring out the exact order of 4 chemical units: adenine, guanine, cytosine, and thymine along a DNA strand. From a chemistry point of view, this is not about reading letters. It's about detecting chemical events and translating them into information. DNA itself is a linear polymer. The sugar phosphate backbone is repetitive and chemically uniform. The only thing that changes from one position to the next is the nitrogenous base. Sequencing works by turning differences between those bases into measurable signals. Almost every sequencing method starts with the same reaction: DNA polymerization. A DNA polymerase extends a strand in the 5' to 3' direction. The 3' hydroxyl group on the growing strand attacks the alpha phosphate of an incoming deoxynucleoside triphosphate. A phosphodiester bond forms, pyrophosphate is released, and the strand grows by one base. Magnesium ions stabilize the charges and make this reaction possible. The key idea is that every base addition is a discrete chemical event. Sanger sequencing exploits what happens if that chemistry is interrupted. Along with normal nucleotides, the reaction includes dideoxynucleotides. These lack the 3' hydroxyl group. If one is incorporated, the chain cannot continue because there is no nucleophile for the next bond. Modern Sanger sequencing labels each dideoxynucleotide with a different fluorescent dye. The reaction produces fragments that stop at every possible position. Those fragments are separated by size using capillary electrophoresis. DNA's negatively charged backbone causes fragments to move in an electric field, and smaller fragments migrate faster. A laser excites the dyes as fragments pass a detector. Fragment length tells you position. Fluorescent color tells you which base ended the chain. Illumina sequencing also uses polymerase chemistry, but with much tighter control. DNA fragments are attached to a glass surface and amplified into clusters so the signal is strong enough to see. The nucleotides are chemically modified in 2 ways. Each base carries a fluorescent dye, and each one has a reversible blocking group on the 3' position. During each cycle, only one nucleotide can be added. After incorporation, the flow cell is imaged, the color at each cluster identifies the base, then the dye and blocking group are chemically removed, restoring the 3' hydroxyl. The cycle repeats. Illumina sequencing is essentially controlled, stepwise polymer synthesis paired with fluorescence detection. Ion torrent sequencing measures a different consequence of the same bond-forming reaction. When a nucleotide is incorporated, a proton is released into solution. In a tiny reaction volume, that proton slightly changes the pH. Templates sit in microwells on top of ion-sensitive transistors. As nucleotides are flowed across the chip, incorporation events cause detectable voltage changes. No dyes or cameras are needed. The chemistry is simple, but the signal grows non-linearly when several identical bases are added in a row, which makes long homopolymers difficult to measure accurately. PacBio sequencing watches individual polymerase molecules in real time. The setup confines light to a nanoscale observation volume, so single enzymes can be observed, even with fluorescent nucleotides present. The fluorescent dye is attached to the phosphate group of each nucleotide, not the base. When the nucleotide is incorporated, the dye is cleaved off as part of normal chemistry, leaving natural DNA behind. Each incorporation produces a brief flash of light whose color identifies the base. Because the enzyme is observed continuously, the timing of these flashes also carries information. Subtle changes in polymerase kinetics can even reveal certain chemical modifications of the DNA. Nanopore sequencing takes a completely different approach. No polymerase is required. A voltage is applied across a membrane containing a protein nanopore, creating an ionic current. A A motor protein feeds single-stranded DNA through the pore at a controlled speed. As short stretches of DNA occupy the narrow region of the pore, they partially block ion flow. Different sequences perturb the current in different ways. The measured current trace is translated into base calls using computational models. Here, the chemical identity of DNA is inferred from its effect on an electrochemical signal. Across all these methods, the unifying idea is the same. DNA bases are never read directly. Instead, sequencing converts molecular scale chemistry to light, voltage, timing, or current changes that instruments can measure. The raw signals are then processed into a sequence with associated confidence scores. Different platforms choose different chemical events to detect, but they are all analytical chemistry techniques applied to the same polymer.